welcome back to the Good Knowledge channel. As the global population continues to grow, can we ensure enough food for over 8 billion people on Earth while maintaining safe and sustainable agricultural practices? And is there a way to boost crop yields without relying heavily on chemical fertilizers and pesticides? The answer lies in advanced biotechnologies that are transforming the way we farm. So, what exactly are these technologies? Let's find out in this video. Gene editing technology, CRISPR/Cas9. The CRISPR/Cas9 technology is ushering in a new era in agriculture, allowing scientists to precisely and intentionally edit plant genes to create new varieties with superior traits. One of the standout applications of CRISPR/Cas9 is enhancing the resilience of crops to adverse factors such as drought, pests, and saline soils. This can help increase crop yields and minimize damage caused by harsh environmental conditions, ensuring a more stable food supply for a growing global population. In addition, CRISPR-Cas9 can enhance the nutritional value of agricultural products, such as developing golden rice varieties rich in vitamin A to help prevent malnutrition. This technology also enables the creation of crops with shorter growth cycles that require fewer resources, thereby reducing their negative environmental impact. However, the use of this technology is still subject to much debate and concern regarding biosafety and ethics. Although CRISPR-Cas9 offers highly precise gene editing capabilities, there is still a risk of unintended changes off-target effects, which could result in uncontrolled traits. This could alter gene function or lead to unforeseen health issues in the edited plants or animals. Additionally, releasing genetically modified organisms into the natural environment could have unpredictable effects on ecosystems. Therefore, the application of this technology requires stringent regulation and oversight by authorities to ensure safety and sustainability in global agriculture. Tissue culture technology, also known as cell cloning, is one of the modern methods widely applied in agriculture. This technique allows scientists and farmers to produce thousands of plant clones from just a small tissue sample, such as a leaf, a stem segment, or even a single cell. What does this mean? It means we can rapidly and uniformly propagate high-quality, disease-resistant, and high-yielding plants. For example, if you have a rare orchid or a high-yield apple variety, instead of waiting an entire year for the plant to mature and flower, you can simply take a tissue sample and clone it into thousands of identical plants in a short period of time. With this technology, we can address issues caused by climate change or pests. When a particular crop variety is threatened by disease, we can quickly clone healthy plants to replace them, preventing crop failures and ensuring stable yields. However, tissue culture requires stringent techniques and a controlled environment, from sterilization to temperature and humidity regulation. But when applied correctly, this technology can yield outstanding results, contributing to sustainable agricultural development and meeting the growing global food demand. Genetic engineering in crops, also known as genetically modified crops, is one of the greatest advancements in modern agriculture. Have you ever heard of Bt corn or herbicide-resistant soybeans? These are all products of this technology. By inserting one or more genes from other species into the DNA of crops, scientists can create new varieties with superior traits, such as pest resistance, herbicide tolerance, or even enhanced nutritional quality. The benefits of genetically modified crops are clear. They help increase yields, reduce the use of harmful chemicals, and support farmers in tackling challenges posed by climate change. However, this technology has also sparked considerable debate. Some people are concerned about potential impacts on human health and the environment, as well as the issue of monopolies held by large corporations in the seed industry. But whether you support or oppose it, there's no denying that genetic engineering in crops is transforming the way we farm and supply food globally. Microbiome engineering is an emerging but promising field in agriculture. Did you know that there are billions of bacteria, fungi, and other microorganisms in the soil and around plant roots? They not only influence plant growth, but can also determine whether a plant can resist diseases. Therefore, by manipulating and adjusting these microbial communities, we can help crops grow more vigorously, absorb nutrients more efficiently, and even enhance their resistance to pests and harsh conditions. For example, scientists can develop special microbial products to promote plant growth, or use good bacteria to compete with and eliminate bad bacteria that cause diseases. Unlike pesticides or chemical fertilizers, which lead to soil degradation, soil pollution, harm to soil microorganisms and animals, and environmental ripple effects. Modifying the microbial ecosystem is much more natural and sustainable because it relies on the natural ecological balance of the soil microbiome. However, for this technique to be truly effective, we need to fully understand the complex relationship between plants and microorganisms. If successful, microbiome engineering could usher in a new era of sustainable agriculture, helping us produce safer and more environmentally friendly food. Biological fertilizers and pesticides are increasingly popular in agriculture due to their environmental friendliness and sustainable effectiveness. Instead of using harmful chemicals, we utilize natural compounds such as bacteria, fungi, or even plant extracts to provide nutrients for crops and protect them from pests and diseases. For example, biological fertilizers not only provide essential nutrients, but also help improve soil quality and enhance the growth of beneficial microorganisms in the soil. Biological pesticides, on the other hand, can directly target the nervous system or growth processes of pests without harming humans, animals, or other beneficial organisms. This brings a dual benefit, ensuring consumer health safety while protecting the ecosystem and biodiversity. However, the biggest challenge is to make these biological products affordable and effective under real-world farming conditions. But if we invest in the right direction, biological fertilizers and pesticides will undoubtedly be a great alternative to traditional agriculture, helping us move towards a greener and more sustainable agriculture. This is a modern method in agriculture and livestock that uses genetic markers to identify desired traits in crops or livestock. Instead of waiting for these traits to become apparent, they can be detected early using technologies such as PCR, a molecular biology technique used to amplify a specific DNA segment in the laboratory, or SNP, a technique for analyzing small genetic variations in DNA. For example, we can develop rice varieties that are disease-resistant, drought-tolerant, or have higher yields, or select livestock breeds that grow faster and have better immunity. However, this method also has some limitations, such as high implementation costs and the need for extensive knowledge of genetics. But with the continuous advancement of science, marker-assisted selection will continue to progress, helping us not only increase productivity, but also ensure the quality of agricultural and livestock products in the future. These applications not only help increase productivity and product quality, but also contribute to environmental protection and global food security. However, careful regulations and assessments are needed to ensure the safety and sustainability of these technologies in agriculture. Don't forget to like, share, and subscribe to support my channel. Thank you so much.